All right, grade 12s, welcome to chapter 17, where we're going to turn our attention to cell division. Now, within cell division, we're going to be talking specifically about mitosis and the cell life cycle. We'll talk about cloning, stem cells, and identical twins. Then we'll look at the process of meiosis and the formation of gametes, gametogenesis. We'll also talk about something called non-disjunction and karyotypes, life cycles of organisms. Here we go, we're going to kick off with mitosis for today's lesson. Mitosis, of course, is very important from day one, literally day one of you as a human being. Remember, you're that zygote, and the very first thing you're going to do is divide, cleavage, multiply yourself, and that's completely done by the process of mitosis. So mitosis is a significant event because it is the making of exact duplicates of cells. So that's going to be important in the formation of for growth, as well as for replacing any dead or dying cells. So the most numerous kind of cell in your body is the red blood cell, of course, and red blood cells have about a 3-month lifespan, and then they are going to die and need to be replaced. So if you remember from our study in grade 11, red blood cells are made in the bone marrow. So the stem cells, which produce all the different kinds of blood cells, need to create replacement red blood cells, and that's going to happen by the process of mitosis. So we're going to define mitosis as a type of cell division in which what we call the daughter cells are going to receive the exact same number of chromosomes as the parent cell. So it's essentially the division of the nucleus. So remember that your DNA is stored in the form of chromosomes in the nucleus of your cells. And so in order for you to produce identical cells, go from one cell to 2 identical cells, you have to be able to duplicate those chromosomes and then pass them from the parent to the daughter. Technically, the parent sort of becomes the 2 daughter cells as it grows and splits. That's exactly what we need to talk about. Also, not only are we going to divide the nuclear material, but we need to divide the rest of the cytoplasm and organelles as well. That's an important process called cytokinesis. Remember that cyto is a prefix that means cell, and kinesis means movement. So it's the division of the cytoplasm. So together, one mother cell is going to divide into 2 identical daughter cells. Something we do have to talk about is the cell life cycle. So for any given cell, it goes through a period of time called the interphase, which is the majority of the lifespan of a cell, where the cell grows, where it does whatever the cell's job is supposed to be, but it also needs to do, as part of this part of its life cycle, it needs to go through what's called its S phase, which is where the DNA gets doubled. Because remember, you have to double your DNA if you're going to split. In this period of time here, the M phase, or the actual mitosis phase, which is only about 10% of the cell's life cycle. So 90% of its life cycle spent in interphase, about 10% spent in mitosis. So as I mentioned, interphase, remember the prefix inter means between, and phase in a sense means the M phase, mitosis. So it's everything that the cell is doing between when it's dividing. So like I said, it's carrying out its normal cell functions, relatively low nuclear activity. Remember we talked about the S phase? In the S phase, we have to do DNA replication. Now, your DNA in interphase exists in the form of what we call chromatin, which is just like, think of it as a very, very, very long strand. The long strand of chromatin actually needs to thicken, and it's going to become what we call as the chromosomes, kind of the packaged version of the chromatin. And I know you guys have heard of chromosomes before. That's the packaged up DNA material into 46 separate packages for a human cell. But a chromosome we're going to talk about this, exists as two sister chromatids. Because remember, we have to duplicate, double that DNA exactly in S phase. I really recommend, guys, that you watch some of the videos that I have in the Google Classroom. It's going to make things very clear exactly in terms of how this works and this vocabulary that you need to know. All right, so if you can kind of see it here, guys. So again, we have a one-stranded chromatid, a certain amount of genetic information that is going to double in S phase. So we have two sisters. Those sisters are going to be joined by what we call the centromere. So the centromere is a region where they are actually bound together. And we're going to talk more about that as we go on. Now, often it's important to be able to count chromosomes or chromatids, and we do that by counting the centromere. So we talked about this thing called a chromatid. It's a single strand of a chromosome, which is going to join its sister by the centromere. So after S phase, each chromosome has one centromere, but two chromatids. Now, we break mitosis up into stages: prophase, metaphase, anaphase, and telophase. Easy way to remember that is PMAT. And if you want to put interphase as part of the whole life cycle, it's IPMAT. But remember that mitosis does not include interphase. It's its own separate stage. And so PMAT form the stages of mitosis. And we need to talk about each one of those. And it's a continuous process. It looks something like this. So these are like electron micrographs, which give you an idea of what it would look like in reality. Here's our artist's rendering. But notice we have taken prophase and broken it up into an earlier stage of prophase and later prophase. Then we enter metaphase, anaphase, and telophase. So we're going to come and break down each stage right now. So prophase. We have our sister chromatids present. So the long-stranded chromatin, which is pretty much invisible in the nucleus, it condenses and to become actual chromatids. So they shorten, they thicken, and then the sisters, remember, are joined together. So there's a sister, there's a sister. We're going to join it with a centromere. Now we have within animal cell cytoplasm, we have structures called centrioles. So centrioles are important in helping the cell divide. They create what are called strands called spindle fibers. And the spindle fibers are proteins that are going to enable the chromosomes to be moved or pulled around in the cytoplasm. So the centrioles, they migrate, there's pairs of them, and they migrate to the poles of the cell, and then they create their spindle fibers. So we talked about the centrioles. Plant cells do not have or need the centrioles. The spindle fibers are protein structures, and they act as guide wires. And they facilitate chromosome migration, or the moving of the chromosomes. So the spindle fibers, they need to attach to those centromeres. Remember, the centromeres join the two sister chromatids. Remember, it's all your DNA. It's just DNA nicely packaged up. All right, so now you see at the end of prophase, by the way, remember that we would normally see a nucleus in interphase, but in prophase, the nuclear membrane dissolves. And here we see the thickening of the chromatin into visible chromosomes. Chromosomes are two sister chromatids joined by a centromere. There's our centrioles at the poles. They've created their spindle fibers, and the spindle fibers have joined at the centromere. During metaphase, our next stage of mitosis, the guide wires, the spindle fibers, pull these chromosomes to the middle of the cell. We call that the equatorial plate. So that's what this slide is essentially letting you know. There's that term equatorial plate. Now in anaphase, so here we are in anaphase, you'll see that the spindle fibers are going to contract towards the centrioles and pull those sister chromatids apart. So each chromatid of the double-stranded chromosome is pulled apart by those spindle fibers. So here we go with our little count. We had 46 centromeres, so we had 46 chromosomes, which gives us a total of 92 chromatids. But now we have 92 centromeres because we've split those sisters, so each one of those sisters has its very own centromere. And we could actually say, now that we have individualized centromeres.
Attention all teachers, I'm sorry for the interruption. We actually have a family tour that's going to be taking place right now. They're going to start in the junior high school wing. So just to give you that information. Thank you.
Might have visitors. So let's see if I can wrap this up before they come into my classroom. But it's just funny, the timing of it all. So we have, as I was saying, since the sisters have split and each one has a centromere, we have 92 individual chromosomes because we want to take those chromosomes and remember, have 46 chromosomes in each daughter cell. So the chromatids are pushed or pulled to the two poles of the cell, and the same number at each pole now is 2n. Now, we're going to talk more about this n representation later, but n stands for what we call the haploid number, which means the different chromosomes that are present in an individual. All right, so for humans, our haploid number is 23. We have 23 different chromosomes, but they pair up, right? One from mom, one from dad, so that we have 46 chromosomes, 23 pairs. So 2n is our 46 number, and n is our 23 number. And they'll be identical at each pole of the cell. So our last stage is telophase. We're going to get our nuclear membrane back. The chromosomes are going to unravel and become chromatin, and we're going to start to get a pinching of the cytoplasm so that we end up with two distinct cells. So the chromatids have migrated, that's already taken place. They begin to unravel or unwind. The spindle fibers are not needed anymore, so they break down. The nuclear membrane begins to reappear, and that technically is telophase. So the last thing that needs to happen is something called cytokinesis, which is the division of the cytoplasm, sort of considered outside of the telophase proper. So together, mitosis and cytokinesis produce two daughter cells from one mother cells, and they are identical. There's a little picture. This is when cytokinesis is over. We have our two daughter cells from what was our one mother cell. This is just a summary slide that I'm not going to go through and read for you, but when you're reviewing, those are the major points that I just went through in terms of mitosis: prophase, metaphase, anaphase, and telophase.